We'll want to know these terms. Activity is, well, first of all, we're focusing on these little red beads. These are the enzymes. And the activity tells you the total units of enzyme in the solution. Total units of enzyme in the solution. Specific activity is also known as a measure of the purity. It's the number of enzymes per milligram of solution, per milligram of protein. Okay, here's a chart. This is a table, and here's the this part is showing the different separation techniques. Okay, so we start with a big beaker full of proteins. Right here, our crude cellular extract, and then let's say we do ion exchange chromatography. It's going to affect the activity and the specific activity, and our goal is to make it more pure. Okay, and so for example, like a test question you might be asked on this, specific activity might not be here. Okay, that might not be here at all, and the teacher might ask you to find specific activity. So my first point is, how do you find specific activity? We just take activity and you divide it by total protein and you get specific activity. Note that the higher the number of the specific activity, the more pure it is. So as we move down, we're getting more pure. And so you could also be asked a question, which separation technique was the most effective? And you'd look for the biggest jump in specific activity. So going from 600 to 15,000 was pretty effective. So that would have been the most effective separation technique. So affinity chromatography was most effective. Okay, here's the levels of protein structure. You have a primary, a secondary, a tertiary, and a quaternary. Primary structure is just the sequence of amino acids. Secondary structure is just the hydrogen bonding. Tertiary structure is going to be the three-dimensional folding of the secondary structures. And then the quaternary is when you have two or more polypeptides together. Looks like that's it.